So I work with this human version of pluripotent stem cells called naive cells that can make those blastoids.
Mm,
And so that's a big prong of work that I'm going to be doing.
that's very exciting. I'll have to come and see your lab when I'm back in Sheffield.
Yeah, absolutely. I was hoping you'd be around because I could come show you like the before and after. What does an empty lab look like, and then what does a functional lab look like?
Yeah, yeah. Um, you talked about how a lot of the research would be done. I think this is a final question for me now because coming towards the end. But I just want to ask about, a lot of the research would be done on mice and stuff like that. How would that look? Was that something you would do in your lab, or was that, would they just have loads of mice on site and how would they get embryonic cells from mice? Like, how does that work?
Yeah, so it's very well regulated. So uh, caveat, I'm very much pro use of animals in research, if it is for a good reason and you're doing it at kind of the minimum need possible, right?
Mm.
And so there's a massive framework in place to minimize the suffering of animals while still understanding that one of the best ways to do this work and certain types of work is through the use of animals.